Okay, so today we have Merritt Maduke, and she is in the Department of Molecular and Cellular Physiology at Stanford University, and she's going to touch on the topic that we had last time again. It's on mechanosensory biology, but I think she's going to give us a different perspective. So take it away, Merritt.
Yes, definitely. Thank you, Kim, for the invitation to be part of this really terrific course. And yes, I'm happy to pick up from where Scott left off and continue the theme of how mechanobiology relates to ultrasound neurostimulation. Oops, why won't my slides advance? Oh, sorry, there we go. Okay, so the outline for my lecture, I'm starting from the perspective of someone who is a molecular biophysicist and wants to understand how ultrasound neuromodulation works. And then I'll tell you about 3 systems that we've worked on in my lab. I hope trying to put it in the broader context of this big question: model membranes, ion channels, and brain cycles. So this is a overview of our thinking. And by our, I mean the team of scientists that my lab has been working with in answering the question, what is the mechanism of ultrasound neuromodulation? It's a very complex subject because between this the ultrasound, which is the initial impulse, that thing that is given, and the output, the behavior, the neural response that happens, there's a huge black box and with many components outlined as shown here. And this is, again, how we've broken it up to help clarify our thinking about this problem. Steve Backus, in his really great lecture, introduced you to this concept of the physical effect of ultrasound radiation. Ultrasound, it could be acting through radiation force or cavitation or temperature, but that's separate from what the biophysical mechanism would be. Is it acting on ion channels or membrane fusion? Or as Scott said, it doesn't have to be an ion channel that's a mechanosensor, it could be something else, but whatever that is, a biophysical mechanism. And then as Steve explained, it gets very complex from that. With even when you have only 2 neurons in a circuit, there's dynamics that come into play. And when you have a whole mammalian brain, it's much more complex. So for today, we're sticking with the molecular perspective, but keeping in mind what we ultimately want to do is solve this entire problem. And to do that, we need people with all of these levels of expertise. And this is just showing the terrific team we've been working with and the systems we're studying. And today, what we're talking about are these simpler systems working on this earlier part of the pathway. So again, Steve did a great job introducing you to the concept, the fact that we can do a pretty good job modeling a neural membrane, but really any cell membrane, as an electrical circuit. It's pretty remarkable how well you can make a simple circuit, a capacitor, which is a lipid membrane. It's really a thin insulator. A 1 nanometer slab of grease is a capacitor in parallel with various resistors, variable resistors, voltage gated sodium and potassium channels, and what we call leak, which is really an oversimplification, that's okay. And then these batteries, each of these variable resistors has a battery, which the main one in biology is the sodium potassium ATPase pump that uses the vast majority of the energy you produce every day to pump sodium out and potassium into every cell of your body all day, every day, day and night, to make these batteries that lets our cells work, neurons and others as well. So that's the framework for electrical modeling of the cell membrane. But from there, I want to take a brief segue, a brief footnote, diversion on this topic. Again, Steve touched on it at the end of his lecture, but I want you to keep this in mind, because with this, although this simple model of a sodium potassium capacitor works, the reality is life is so much more complicated than that. And this is that there are thousands of ion channels. This is a really simple overview. You've got chemically gated ion channels, electrically voltage gated ion channels, mechanical and thermally gated ion channels. This slide I still is a little bit old because it didn't even include potassium selective ion channels as mechanical, but now we know there's very many. And within each of those, there's non-selective cation channels, chloride selective, calcium selective, potassium selective. So this is a morass of ion channels. And to give you even a more of a picture of how complex it is, if we take just one piece of this, potassium selective channels, and zoom in on those, there's, oh, this is a little fuzzy, but I think you can get the idea. There's 4 or 5 major classes of potassium channels, all very different, some voltage gated, some ligand gated, some mechano gated, temperature gated, and they're in different types of families, different. And within these 4 or 5 families, however you count it, there are, in a human body, there's 80 different types. So it looks like 5 here, there's 80. And that is not counting the many dozens of splice variants on each one. So if you start to do the permutations and combinations, it's overwhelming. So just keep that in mind when the simple models are to help us design experiments in an interpretable manner, but ultimately biology is very complicated. And so if this sets up my problem, which is, yeah, we can model the cell as a circuit, but really to be totally accurate, you'd have to add many, many more resistances, and it would go on for hundreds and hundreds more. So that's really complex. And when you're a biophysicist trying to answer the question, simple question, what's the mechanism of ultrasound neuromodulation? And you have to think about all these hundreds or thousands of possibilities of interactions between ion channels and biological molecules. It becomes a bit daunting. And so what our approach has often been to let's realize that the problem is complex, that let's step back. Wouldn't it be great if we could develop a experiment, a simple experimentally controllable system where we just study a piece of these and where we can really carefully control the experimental variables and therefore be certain of our interpretations before developing a model in the more complex reality of life. So wouldn't it be great if we had such a system? Well, we actually do. So yeah, this slide is just about what I said. How do we get a simpler system to address this question? And the simplest system I can think of is just a model membrane. And the technique is called planar lipid bilayer recording. And it's really remarkable that this can be done. So here's a diagram. It's just two solutions, aqueous solute, biological buffered solutions, separated by a divider. And in that divider, there's a small hole. And what the remarkable property of lipids is that they spontaneously form bilayers. That's an energetically low structure. That they don't need, enzymes and stuff to do that. They spontaneously form this. So if you paint, take a solution of lipids in an organic solvent and just paint them across a partition like this with water on either side, buffer, they will spontaneously form a bilayer. And now you have an artificial membrane with no proteins, just the membrane. And you can stick electrodes on either side of those, hook them up to an amplifier, and now make recordings of, well, in this case, it would be the capacitance because there's no resistors, just the capacitor. We've got a very simple model of just the capacitor. And what's even better about this system is that you can take proteins one at a time. Say you wanted to study a potassium protein, purify it away from the membrane and add it to this membrane. Membrane proteins spontaneously like to go into membranes, and so it will insert spontaneously. And now you have one type of ion channel to study in isolation in a simple system where you can ask very specific questions. So this was when I first heard about ultrasound neuromodulation. I'm an ion channel biophysicist, and actually I should tell this story to inspire Stanford students around here, which is you picked a great place to be at Stanford. It's a place where teams of people can get together very easily because there's so much enthusiasm about forging new ground and interesting questions. And Bill Newsom introduced me to Kim and got us working together on this project, which is really, really exciting. I never would have on my own thought to look at ultrasound neuromodulation, but I did when Bill brought up the question. I thought, well, I don't know how it works, but I know how I would start to study it in a simple system. So let's try it. So that's what we did first. Now here's a cartoon diagram of what a bilayer recording system looks like. And actually, so ours is, I showed you a vertical example, that's more standard, but for technical reasons, we have a horizontal setup, but they work the same way. The idea is you have a partition between the two chambers. You still have electrodes going to amplifiers in both chambers, and you paint a lipid bilayer across the chamber. Here's an actual picture of one of our chambers, the one that was used with ultrasound neuromodulation. It's just milled out of Delrin material. Here's the top chamber. You can't see the bottom chamber, but this electrode here is going through to the bottom chamber, which is underneath, and this electrode is in the top chamber, and they're in the amplifier. Here's our ultrasound transducer hooked up to its own amplifier, and everything's controlled together. So now you can find out, what does ultrasound do to membranes? This, I should give credit to this. All of this work in my lab was pioneered by a really wonderful postdoc, Martin, who's thoughtful and got things to work in just really in a wonderful way. So these are all his experiments. And I have to say, the first result, which is what you're looking at here, was not what I expected. So this is voltage clamp experiment that Steve told you about, holding the membrane at minus 200 millivolts and measuring the current. And normally what you would see with a membrane, there is no current because membranes are very tight. They're not really, they have very little ions leak through membrane, so there's no current. But when Martin applied ultrasound, he saw a huge current. 200 picoamps is a huge current in a bilayer. Often in this type of experiment, we are usually measuring single channel currents on the order of 1 or 2 or maybe 10 picoamps. So seeing this enormous oscillating current in response to ultrasound, no ion channel there, was completely surprising. Did not expect this, but we'll get, but we should have, and I'll explain that in a minute. But first, let's get at this question, the physical effect. As Steve told you, radiation force, temperature, and cavitation have very different frequency dependencies. 1 megahertz, well, any of those could be happening. But if you go to a higher frequency, say 43 megahertz, that's a non-cavitational frequency. So if you still get a response, it will be radiation force or temperature. And that's what Martin saw. So here's a megahertz and 43 megahertz, you see actually a stronger response at 43 megahertz. So, so this is a non-cavitational mechanism, and as I'll argue going forward, it's a radiation force mechanism. But first, back to this question, where are these enormous currents coming from that I didn't expect? I expected to see like some little current, and then we would study the ion channels. So, but I should have expected it. And you probably have already thought of this based on Steve's lecture earlier, where he pointed out that the capacitive current, which is just the derivative of the charge, is C dV dt plus V dC dt, but capacitance doesn't change, so we normally ignore it. But if ultrasound is changing the capacitance of the membrane, you should see a current. And what I didn't realize, and we did the calculation, actually a very small change in capacitance, less than 2%, would be expected to produce this current. So as soon as we did that calculation, we thought, oh yeah, of course. And one other thing I should say, you know, it can't be a leak current because it's going positive and negative directions under voltage clamp. So if it was a leak current, it could only go positive or negative, not both ways, held at minus 200 millivolts. So this is V dC dV, it's a capacitive current. Okay, so the current must be capacitive. That means the capacitance is changing. How can that happen? So in reminding, we're C dV dt, we cross out. In this case, we cross this out because we're under voltage clamp. Voltage is not changing, only capacitance. So the current is V dC dt. And what is capacitance? It's the dielectric constant of the medium times the area over the thickness. That's pretty simple. So either the area or the thickness of the membrane must be changing. Oh, my phone thinks I'm talking to it. Nope.
I didn't understand that.
What's happening here? So to interpret this in terms of the experimental setup, we need to think more closely about what that is. And as I told you, this is a horizontal bilayer setup. And actually in our standard experimental conditions, the bilayer is not really flat, it's a little saggy. And that's just because of the hydrostatic pressure of the way this experiment is set up. So the bilayer under resting conditions is a little bit curved downward. So what we're imagining is ultrasound's coming from underneath, and as the radiation force is pressing on it, then it should be pushed upward. And if you look at that, that makes sense. That means the area is getting smaller. That fits with this signal. So the initial current is positive. That's the voltage is negative. The current is positive. That means the DC dt is negative. The area of the bilayer is getting smaller. And if you integrate this overall, it's getting smaller. That makes sense with the setup. So what we're saying, why is it ringing? Well, it must be, this is not a cell that has tethers and cytoskeleton and everything. It's just a membrane. So the ringing makes sense too. You've got this, just a lipid bilayer sitting in a curve, and ultrasound radiation force impinges on it and it pushes it upward. And then there's a ringing until it settles down to a new capacitance level. And then when you remove that pressure, you get the ringing in the opposite direction as it relaxes back down to its original state. And I'm not going to show you the data. Were there a question?
Yeah, I have a question. You usually think of lipids as being less dense, and why would they not float? Why is the resting membrane not sort of pointing upward? Why is it sagging?
Well, it's just because we have the volume of solution here where we set it up it's the hydrostatic pressure from the solution below. And what I'm not showing you, but it's in this paper, is if we change that, if we put less solution here, we get a flat, or we can get it curved the other way, and we can change the direction of the signal. So that was a good test of this model that we're thinking of. But the even more impressive test that I will show you, or I think is more impressive, is this idea, okay, if that's right, if this bilayer is moving up and down, then we actually, Pierre is the one who said this, Curie-Yacoub, hey, well, if it's really moving, we should be able to detect this with laser Doppler interferometry. And so again, the great thing about Stanford, Martin put his whole electrophysiology rig, amplifiers and ultrasound transducer on a cart and rolled it across the street to engineering to use laser Doppler interferometry in Pierre's lab. And all basic is measuring the time of the kinetics of the laser reflection from the surface to see if the surface is, the distance from the laser to the surface is changing over time. And it is. So on top is the ultrasound experiment signal that you've seen before, just on a different time scale. And on the bottom, that's on the bottom. On the top is the laser Doppler interferometry signal. And you see the velocity, the bilayer moves away and towards and away and towards until it settles down to a new level. And then when the ultrasound's off, the laser detects the bilayer moving again. And these, if you overlay them, they're perfectly matched up. So that interpretation of the ultrasound impinging, pushing on the membrane is supported by this experiment. And so the conclusions from these studies is that ultrasound radiation pressure, so measure, distorts the bilayer structure, causing changes in capacitance and membrane curvature. So that, I mean, that was a very satisfying study, but of course you want to know what happens in cells. And my idea of testing out, well, two things. One, the idea of looking at ion channels in the bilayer system was made difficult by this huge current from the capacitance change. It would be hard to see the ion channel currents underneath that. But also the thing we realize is this is, I mean, there's surely a huge radiation force pushing on a bilayer. But in a cell, as I mentioned, there's cytoskeleton things holding it down. It's going to be different. So it's not going to be, we don't expect it to be so floppy, but we want to know, is there effect? Is there a pressure on the membranes that influences things in the cells? And the idea is just that there's the radiation force pushing against the cell membrane wouldn't make it have these huge displacements, but should put some tension on the membrane that may open ion channels that are gated by mechanical forces. And so to test this idea, we moved to a model cell system. Again, not a neuron, not even a neuron, just a cell, HEK cell, which is a typical tissue culture cell that is excellent for patch clamp experiments, and because they're really good at expressing proteins, ion channels for whatever DNA you put into them. And so this experiment is set up pretty similarly to the bilayer experiment. There's instead of having two chambers, we just have one, and you place the cell on top of the chamber, on top of a thin polystyrene film, basically to not absorb too much ultrasound and create heating, and do a patch clamp experiment where you put the pipette on the cell and measure currents of whatever channel. And I'm giving you a hint here, piezo channels are what we're looking at. As an overview, there's more or less 3 types of mechanically sensitive channels. Of course, with the caveat that anybody who studies either mechano or thermosensation will tell you that all ion channels, all proteins, all life is sensitive to mechanical and thermal stimulation. So everything is. We just call these mechanically sensitive because they're very, very sensitive. Mescal you heard of from Scott. Remember that was a bacterial pressure valve one that when you put it works by a stretch mechanism. Trek 1, Trek 2 you heard about from Scott also, and that really beautiful mechanism of it, of the enzyme and the lipid moving from domains might be give the mechanosensitivity. Piezo 1 is kind of the mother of all human mechanosensitivity. Look at it, it's huge, and it just look at the shape tells you. And my personal opinion is it probably works like a hybrid mechanism. It's just you can just looking at it, you can say, oh my goodness, stretch is going to open that channel, right? But it's also very sensitive to lipids and may have some of this domain movement mechanism going on. I don't know, but what our question here was, is piezo 2 responsive to ultrasound? So to test that, we expressed piezo 1 channels in HEK cells and measured currents. And what you're seeing here, so this is ultrasound at 50 watts per centimeter squared, and we're measuring current. The black trace is a typical piezo current, is voltage clamp at minus 80 millivolts. See, an inward current is a non-selective cation channel current that desensitizes, deactivates very quickly. It's a classic piezo current. And it's induced by ultrasound. We didn't poke the cell, we didn't do anything, we just looked at ultrasound, a big current. Now, what's interesting about these experiments is actually, if we applied ultrasound when the cell was just adhered to the cover slip, there was no response at all. That's this gray tracer, nothing. And in fact, we thought, well, maybe because it's just adhered, these are adherent cells, maybe because it's so squished down on the surface, it's not responsive. So let's detach the cell. You can do that easily in patch clamp and just raise it to the ground level. But even when it was detached, it had no response. It only responded when it was lifted up a little bit. This is going to make sense in a minute. So these are just summary data saying if it's lower than 50 microns, no response, and then raised to 50 microns to get lots of piezo 1 currents. And it's reversible. You can go up, or down, up. Very robust response. So ultrasound is activating these mechanically sensitive piezo channels, but in a way that at first, why does the height above the chamber matter? And it was puzzling because we know as you go further away, further above the cover slip, the radiation force should decrease. It doesn't decrease that much, but it decreases. So if it's a radiation force mechanism, it doesn't make sense that you need that. Until, again, with wonderful collaborators in electrical engineering, they modeled not just the radiation force, but also the streaming velocity. What's streaming velocity? It's a radiation force response of water aqueous solution being pushed by the ultrasound's radiation force. And the reason it has this height dependence is that there's still like a quiet undisturbed layer. I think there's a better word for that. Near the surface, and streaming velocity increases as you go away. So that's what, if you remember what Steve said, what mechanically sensitive cells are going to respond to is not just the pressure, but the strain. And if you can see that in this movie here, yeah, there we go. So there's the ultrasound. Did you see that? So let me play it again. Let's go back. So the cell's just sitting there on the patch pipette, and the ultrasound, that's just pushing on it. So you don't see that when it's lower, only when it's raised up. And these just shows that the hydrodynamic pressure actually gets lower as you move away, and the temperature gets lower. So streaming velocity is the only thing that correlates with this effective increase in current as you move up and away.
So Merritt, in that video, is the streaming, I guess, is the water coming towards the pipette and sort of pushing all the edges?
Yeah, it's coming. Yeah. It's kind of hard. I mean, the picture looks kind of 2D. So the pipette is here and the cell is here, and the ultrasound's coming from below, but it's not. I mean, it's the right. Yeah, it looks like it's coming from the side, sort of, but it's probably it's an angle. That's a good question. It's hard to visually change these things from 2D.
Kim and I we've had deep discussions on this video of yours for like a long time. I think even in this course. And we were always wondering why it collapses from all directions, seemingly the same if ultrasound's coming from, one side or the other. So it makes.
No, I think it's the streaming. The streaming flow is much wider than the cell. The cell's only 10.
Okay, I see. Yeah. Finally cleared it up for us, Kim.
Hopefully. Yeah. So, and well, yeah, we'll see that later on. It was, even when you have a focused ultrasound, the effects are much broader than the focal spot. We see that in our slice work that I'll talk about next. But first, just was sort of a control. It's just looking at another channel. This is a famous NAV channel, voltage gated sodium channel that Steve told you about. It activates very rapidly inward current as sodium goes in, and inactivates very rapidly. And we looked at this, and you can see that ultrasound actually has a little bit of effect on the kinetics. It speeds it up, probably through a temperature effect. Temperature speeds up everything, but this has zero dependence on the height. So ultrasound is activating a mechanically sensitive channel, classically mechanically sensitive piezo 1 channel, but not NAV1. So again, these results are, these are not that important. These are not so important, just a detail. You can read it in the paper if you care. Ultrasound is activating acoustic streaming, which results from radiation pressure, activates piezo 1 mechanically sensitive channels. And so from there, of course, we're using simple systems, but you always want to bring it to the next level. And what we really want to know is this going on in tissue too? And so the solution, right? You're not going to have acoustic streaming in tissue. It's not a bag of solution, but what you would have is, in its place, is displacement and strain, as Steve explained very nicely in his lecture. And so the hypothesis that in tissue, this US ultrasound-induced mechanical strain will activate channels by displacement and strain. And this is just a slide that's similar to what Steve showed on his data, reminding you that what he saw in this multi-electrode array experiment is that the cells would actually physically move. You would see them displace in response to the ultrasound. So that's the idea that maybe that, and this is a retinal slice. And of course, people always ask, well, retina, brain, are they really the same? And Steve makes very good arguments that this is a model circuit that's really a good one to study, but we said, let's study another one. And so we were looking at hippocampal brain slices. So the setup is actually identical to the one we use for the HEK cells, only instead of putting a tissue culture cell onto the surface, we use a brain slice. And I should have put a note here. This is thanks, again, to a wonderful collaboration from Dan Madison, who's a world expert in looking at circuitry and hippocampal brain slices, and taught Martin how to make these slices and record from them and so forth. And they're really, terrific because they have these well-defined layers of neurons, and they've been very extensively and well characterized for 30 years. So what Martin did is, let's walk through this slowly. These are current clamp experiments. So now he's injecting current and looking at how the membrane voltage changes and what happens to action potential firing. So in the top left here is an example of a hippocampal pyramidal neuron into which Martin has injected current, and he sees spiking, just like what Steve described. Cells can spike, repetitive spontaneous spiking of a cell. That's pretty classical firing in a neuron. And what he does below, and always in these slides, ultrasound will be in red. He applies ultrasound starting 500 milliseconds before the application of the current pulse and extending 500 milliseconds afterwards. And what he sees in this example, this one representative trace, is that the firing rate has decreased. Ultrasound is inhibiting action potential firing. And not only in this example, but if you look at interspersed trials, these are control, ultrasound interspersed, we see that on average, after many cells, this is a raster plot. I think Steve introduced some. You're just looking at every time you see a point here, there's an action potential fired. And you could see that overall, ultrasound really inhibits action potential firing compared to control. And here's summary data showing that it's statistically significant. So this is, and I should say, this is a really robust experiment. This is from 24 cells for this example, but Martin has seen this over dozens of preps over many months. Every single time, it's really a robust and reproducible response. And I say that because the reason we went to brain slices in the first place is Martin had spent probably 3 months on cultured neurons. That was the next step after cultured typical cell cultures, was cultured neurons, trying to see a response. And it was not, once in a while he would see a response, but usually he didn't. And we tried all kinds of troubleshooting to say, what is the experimental variable that we're not controlling for here? Why can't we get reproducible results? And finally we just said, Dan, help us, let's look at the tissue. And it makes sense now. Neurons in culture sitting on the dish are right here in this quiet zone where you don't have very much tissue strain, as Steve showed in his slides, or streaming velocity. But in a slice, the slice has a thickness, so they're sitting somewhere up here. And we see this very reproducible response. So it fits with our idea, at least, that ultrasound is pushing on the tissue. And I should say that we're using 43 megahertz here, so it's not a cavitational frequency. So we're thinking it must be radiation force or temperature.
Well, Merritt, if it was temperature, would there be any difference as it's lifted up off the bottom?
That's right. So the temperature, if we could do a distance dependence, the temperature should go down as you go further away. But we can't, in a slice, we can't do that, right? So ideally you'd take the slice and patch at a low position of the slice and then higher and then higher. And that's not practical experimentally. So we couldn't do that test. But as you'll see, we actually have not yet fully distinguished between temperature and mechanical effects in this experimental setup. And that's what I'm going to talk about for the rest of the time here. So I'll go into that in a bit of detail.
Merritt, have you also given thought to other brain regions that may have less or more fluid compared to?
Oh, no, that's a good question. Which brain regions would you compare?
Oh, I don't know. I mean, even just something like gray matter versus white matter regions and axons versus cell bodies. I just imagine if there's more fluid and streaming velocity is responsible, you might see more
effect. That's a really good question. We haven't thought about that yet. We will.
Lots of space to explore with this prep. It's very cool.
So, and this is just the same thing. This is a repeat, elevated cell. The idea is that as you elevate, you'll have that pressure displacement strain in a bigger degree. So the one thing to me that was really satisfying about this study, so I showed you that it's robustly, gives a robust inhibitory response. And this question was of interest because if you read papers in the literature on neuronal excitability and ultrasound, some papers say it's inhibitory. We agree with that. Some say it's excitatory. Some say inhibit. And it's really hard to compare them because they're all using different ultrasound parameters. They're all using different experimental preps, different conditions. It's really impossible to make a quantitative model of what's going on because there's so many differences between these published experiments. So what Martin did next was extremely clever with his idea, not mine. And that was to look at the frequency input curves, to look at different firing frequencies within the same hippocampal slice. And you can do that by just injecting different amounts of current, mimicking different incoming signals in the brain. So in this example on the left, it was probably around 40 picoamp injected current. And you see here, in this example, 40 picoamps is inhibitory. If you go low enough, you wipe out firing entirely. But if you go high enough, this one seems not much is going on. But here in this 250 picoamp example, if you look closely, look at all those red spikes. The firing rate is increased by ultrasound. This is the exact same cell, not just the exact same ultrasound parameter, exact same ultrasound transducer, parameters, cell, temperature, buffer, everything. The only thing that's different is the injected current and the firing rate. And so these are anecdotal representative traces, but if you look at summary data, or actually these are not summary data, this is a frequency input current from a single cell, but you'll see the summary data all look like this as well. At low firing rates, ultrasound is inhibitory, and at high firing rates, it's always excitatory. So this makes sense, or this explains why people have seen different things, because you can easily, even in one, we change one thing and we go from excitatory to inhibitory. So now we have a framework for thinking about this. And actually, this was one of our biggest clues as to what ion channel, maybe mostly, if not all, probably not all, nothing at all, but mostly responsible for these effects that we're seeing. If you look at inhibition, what you can say on the simplest level, well, okay, if you're getting inhibition of firing frequency, you could be activating a potassium channel. As we know, potassium will go out of the cell, make the membrane more negative, that's inhibitory. Or you could be inhibiting a sodium channel. That would reduce firing frequency. You can't tell, either one could be happening, and other things too, but those are the two main players in action potential. But if you go up in firing frequency, then the picture changes. So if you were inhibiting a sodium channel, you should still be inhibiting action potential firing at high firing frequencies. There's no way around that. But paradoxically, activating a potassium channel explains these data as well. And that's not intuitively obvious until you get into the nitty-gritty biophysics, which I won't do too much. But basically, I think Steve told you that. So remember, sodium channels inactivate very rapidly. And they only recover from that inactivation once you go to negative potentials again. What is the thing that lets you go to negative potentials? It's potassium channels. So actually, in this case, potassium channels can help, because if you activate potassium channels, they bring the negative potential back a little faster and let sodium channels recover so you can keep firing. So this one hypothesis, potassium channels being activated, explains both regimes of this ultrasound effect. And Martin did many more tests of this idea by looking at the action potential shape and so forth and everything, and that's in his paper. I'm not going to go through those details, but let's think about this. We call this the potassium channel hypothesis. And actually, it's even more refined to that too. We call it a K2P potassium channel hypothesis. And that's because I didn't make a slide, but what K2P channels are is they are non-voltage dependent potassium channels. And so as you know, voltage dependent channels change their properties as the voltage changes, and voltage is changing like crazy when you're firing action potentials. What Martin showed is that these effects were identical whether he applied the ultrasound at the beginning of the pulse or the middle or the end, anywhere. So what that means is this potassium channel is not changing. It's exactly the same, responding the same way no matter when you get the pulse. So that makes our hypothesis centered around not only potassium channels, but voltage independent potassium channels. And that would be the K2P channel family, which you've seen before. Guess who's in there? Trek 1 and Trek 2 and all these other ones. Actually, it's interesting what this family, I think the first one is called TWIC, and it stands for something like 2p type domain, potassium channels, and Trek is TWIC-like potassium channel, and TASC is TWIC-like acid-induced potassium. Those are really crazy names. Anyway, you've seen Trek before from Scott. It's the K2P mechanogated channel. So that really fits. I mean, we weren't thinking of Scott's work when we did all these experiments to conclude that it must be activating a potassium channel, that's voltage independent, but it fits perfectly. And so from that we say, okay, perfect. Our mechanically sensitive hypothesis is carried on. I need to leave you with one big caveat, and that's what Kim alluded to. Temperature and radiation force have the same dependence when it comes to frequency, and it's often hard to distinguish them. So as an experimentalist, we like to measure temperature, and that's really challenging with ultrasound. Typically what we would use is a thermocouple, but the ultrasound PR tells us, no, you can't use a thermocouple. That won't give you accurate. You need to use an IR camera. Well, we tell them, and he said, but you can't have water around with the IR camera. It's like, well, we're biologists. You're going to have water around. So that leaves really the best way for us to evaluate temperature in the system is simulations. And Pierre's lab has got very good parameters for accurate simulations in different experimental situations. So if we simulate the temperature for our brain slice experiment, we can, in that transducer and power level we use, it actually predicts a substantial 1 degree, not killer, but significant temperature rise over the slice, and actually over a much bigger distance of the slice than the focal point of the focus of the ultrasound. And so we can't rule out temperature effects. And he also modeled the displacement. There's a significant displacement. I don't have a lot of time, and I don't want to get, I was trying to decide how much detail I'm going to. So we know the temperature is probably rising by 1 degree. Trek channels are very sensitive to temperature as well as mechanical. So this temperature would be enough to probably activate some Trek channels. But there's also this displacement. And turning displacement to actual strain requires numerous assumptions about Young's modulus of the tissue and all the properties of the tissue. And we spent a lot of time trying to make unbiased models of what the force would be from this displacement. And I think it's actually, this displacement is causing a force also in the range that will stimulate Trek. And how we can tell that is what you would expect if there was a mechanical force, if this displacement modeling is correct, we might be able to measure that capacitive change. And actually we do. This is the last piece of data I'm going to show. Oops, my little picture's in the way. So this is zoomed in on the very beginning of those currents. Like the spikes would be way out here, way over out to the right. This is the very beginning of the trace. And what you see is this is the ultrasound applied. It's a little tiny rise in the voltage and then a decrease. This tiny rise is a very small one, but it's statistically significant. You only see it when you average many traces. But that's due to a change in capacitance. The ultrasound is affecting the capacitance of the membrane, and it's a fast change. So it can't be an ion channel. This is the ion channel, the membrane potential decreasing. The potassium channel gets activated, the membrane potential decreases. And this is the kinetics of this are on the order of what you would expect from an ion channel response. The kinetics of this initial blip, and you see there's an off blip too, which would be required for a capacitive response, is too fast for any ion channel thing. It's in the time course expected for a capacitance change. So this capacitance change together with this displacement will give you 0.1 to 1 millinewton force that would be enough to activate a Trek channel. So there are a lot of assumptions going into that conclusion, but I personally think it's right. The caveat is that there's also a temperature change for sure, and Trek channels are sensitive to both. So we really need to design some experiments to tease apart whether you can't get displacement without temperature change, or which one of these is more important, or are they both important, or doesn't matter, and what do you want to do in vivo? So those types of questions, I think. So anyway, so there is a temperature and a mechanical effects, and with that caveat, our working model is that ultrasound radiation force causes mechanical change that can certainly modulate ion channel activity in both model membranes, retina, and hippocampal neurons. And with that, I can end and take any more questions you may have.
Oh, that's great, Merritt. Thank you so much. It was really nice the way you sort of reinforced a lot of the concepts that we've talked about already.
Thanks.
Let's see, Keith, do you have any questions?
Well, I was going to ask originally just the methods question. How is it when you develop the painting a bilayer membrane across that prep?
Yeah.
How do you do that? I know that's a weird question.
Yes, I love that question. I know, it seems like magic to me. I mean, I didn't develop this, somebody else developed it. But so how you do it, actually, the way I was taught was you take a little solution of lipids and dip a glass pipette into it and wipe the glass across. That works really well. But what a postdoc in my lab discovered that works even better is you take a pipette tip, and you know how you can, if you suck up and you can blow out and blow an air bubble on the pipette tip. So coat the lipid on the air bubble and drag the air bubble across, and that makes the best bilayers. So
it's a beautiful demonstration of how creative scientists can be. I had one more question, Merritt. This is sort of a grander question, but I think you did a nice job of painting the picture of all ion channels being mechanosensitive. And of course, everyone has only some bandwidth to work on channels that they think are going to be important.
Yeah.
Do you have any sort of grand vision for how we can examine ultrasound's effect on all channels and create some sort of major comprehensive model, or are we always going to be going piece by piece?
No, I know. I mean, ultimately, we'll have a major comprehensive model. I think that's part of the goal of the Allen Brain Atlas. I mean, really just documenting what's there. So I have hope that we can do that. That can happen in our lifetime, even my lifetime, which will be shorter than yours.
So with the Allen Brain Atlas, are all regions of the brains have different ratios of all of these ion channels? Is that correct? Or
yeah, and I mean, you can look them all up. So again, I'm not going to get there in the next 2 years, that's for sure. But I think it's doable, especially with computation becoming as powerful as it is, and so forth. But yeah, you're right. We can't go one by, like, these experiments. I can't go, if I went one by one through ion channels at this rate, it would take the next few centuries, and we don't want to do that. So we want to develop different approaches that include modeling and so forth, and with our experiment. So the idea is to develop principles based on experimental study of representative groups, and then apply them to the bigger picture.
I see. So maybe moving more towards in silico computer-based approaches once you know certain trends.
I like that idea.
Okay, I was thinking maybe Merritt's going in the direction of a super robot to replace Martin.
No, well, yeah.
Maybe.
Automation. Yeah, I mean, Martin had to troubleshoot so many things. That's what a great scientist does. But yeah, once you get past that, automation can help a lot, for sure. All right, thanks for letting me be part of this great course.
All right, thanks so much, Merritt.
Right.
Yeah, great. Let me, yeah.